Okay, so I'm going to talk about sorghum in relation to the description of the phenotypic traits we are using, as we really believe it's a key step to improve our abilities on our studies in terms of genetics on breeding. So my talk will be articulated around three main parts. First, I will share with you my expectation regarding the description of phenotypic traits. In the second section, I will try to identify what I need to efficiently describe my trait and make them useful not only for me but also for the community. And in the last part, I will do some proposition in order to optimize the development of cultural vocabularies and extend their use. Also, some of them are already running. So first, what do I expect in relation to the description of phenotypic traits? So I'm a geneticist. I'm involved in breeding programs. So my goal is really to develop varieties that fit the expectation of the end users for the different end users and growers. So basically what I want is to predict the phenotype of my varieties in different contexts. So basically when I look to a plant, what I want is to decompose it in the main effect of the seeds I'm using, of the weathers I'm going to face, of the crop management I'm going to apply to them. And also I want to track down the interaction between these main terms. So if I put that in equation, actually as a geneticist, I'm really going to focus on the green boxes, which are the genetic components. So what I want really to do is based on my data, on the data of others, I want to estimate the G components and then predict the phenotypes. And I want to predict the phenotypes in non-tested environments that I did not explore for tested and non-tested genotypes. So how do I try to do that? So actually I'm trying to dissect the components of the genotypic effects. So what is the genetic effect? Actually it's the sum of the effect of the genomic regions that control my trait of interest. So it can be large genetic regions as QTLs or really a specific substitution of nucleotides. It can also be some variability of expression between genotypes. And we are also to take into account the interaction between the genomic region. And what is great with genetics is that everything of it changes with the developmental stage. So we also have to track this. So what I really want to do is to identify this genomic region, track the gene expression networks that are evolving during the plant development. And with these two information in hand, I should be able to estimate the breeding values, the genetic components here. And based on that, if I'm working well with the agronomists, modelers, and crop managers, I should be able to predict my phenotypes. But for this to work, I will need to aggregate multi-environment trials and possibly data sets from others. So indeed, if I have only one trial, I'm going to be have a really low efficiency to describe my G components. But if I have N of them, I would be much more efficient to do that. So myself, as a plant geneticist linked to breeding, I only have access to, let's say, 4 or 5 trials each year, which involve my genotype of interest. But as I belong to a community, I can have access to more. And if I'm talking about my species, or even better, the species that are phylogenetically related to my one, I can really increase the power of my analysis to reach that. But once again, for this to work, I need to have accurate description of the traits. So, just an example to go back to the publication. So François already talked about plant 8. We were talking about plant 8 just earlier. So in sorghum, if you look to plant 8, in some publications, the plant 8 is the distance between the base to the tip of the panicle. In other, it's the base of the panicle. In other, it's the base of the peduncle. In other, it's the 8th of the last ligule of the last ligulated leaf. So when I'm talking about plant 8, it's not one trait, it's at least 4 traits. I have the same example for the lignin content, where it's a bit more complicated because we have different methods to evaluate that. These methods are validated, accurate, they are named. But if I look to the correlation of the estimation of these different methods in relation to the digestibility of the forage, which is a trait I'm looking for, you can see that the correlations are really different. So one trait means 3 traits. And if I change the unit, I have new correlation. So one trait is 6 traits. Okay, so complicated. I follow up with the lignin example. So as a geneticist, I want to identify the QTLs that control my traits. So Emma Mace, here, she did a really nice work. She aggregated 150 QTL on GWAS studies since 1995. So she has a total of 200 traits for 6,000 QTLs. So we have this in the database. I like lignin, so I want to look for. So there are 8 genomic regions, great. I have heaps of information. I know the population where it was mapped. I know where it fits. I go to look to the trait, and actually I have no information regarding the method that was used. I have no information regarding the ontogenic stage. I have no information regarding the unit. So, of course, I want to see the paper. Actually, in the paper, none of them are clear. These guys works well. It's published in Tag or something like that, but we have no information regarding these traits. So we really need to define the traits. So what do I need in order it works and makes the trait useful for me and the community? So we have nice guys working on that, the guys from the Crop Ontology, and they developed a relevant framework to describe a variable. So Laurel already described that. I'm not going to bother you with that. So there is entity attribute, then the method and scale. Just one example here. So with the Crop Ontology, we have really a relevant framework to describe our traits. However, we really need to link this information to the other ontologies, as Elizabeth and Laurel already showed. So it's just one example of the traits we are tracking in our experiment. So it's a bit long. It's a lignin content based on this method in the Internet 18, measured at 120 degrees after the conjugation start, blah, blah, blah. So to be able to describe it accurately, we need to go to the plant trait ontology and look for lignin. We find it, that's great. We need to look for the internode. We find it in the plant anatomy ontology, so it's working. For the stage, we do not find exactly our stage, but we have the framework to mention it. We can also have access to the ENVO that we are going to use to describe the 120 degree here. And we have the unit ontology. So we do not find exactly this one, but it's close, so it's not a problem. But the main problem is with the method. I've written here no ontology. Actually, I'm wrong. There are ontologies for biochemistry method, but I was not able to find this one at least. And so I was not able to describe which method was I used, neither describe if it was a prediction, if my result was a prediction of a reference value. And I was not able to tell you what are the kilograms here. It fits dry matter of the stem or cell walls or stuff like that. So we really need to aggregate the crop ontology with the other ontologies to be efficient. So once we have that, actually the pipeline is quite clear. We can start from the crop ontology. I pick one example of plant 8. We can create alias, let's call it 8, and we can enter it into the field book, as we use the KSU software, which is really nice. We can easily transfer it to local database, go to the publication, and as we have that, we are able to aggregate and increase the power of our analysis. So we have a really nice framework. We have all the pipeline is set up, but for me, at least for sorghum, I'm not talking for other species, we really miss the useful content. So I'm going to explain. It's not because it's bad, it's because it's incomplete. So in sorghum, we have 174 traits, variable. Actually, along the period 2017-18, there has been 50 articles published in sorghum genetics and phenotypes. Only one make a reference to that. So it's not useful currently for the community. So we really need to improve the referencing of the published traits. I don't know how it's working in other species, but for sorghum, it's just the beginning of the story. So 3 propositions. Actually, some of them are not anymore valid. So first, we need to fit the crop ontology. I see two ways to do that. It can be individual inputs. I have the framework based on the crop ontology. I set up my trials. I mobilize my colleagues who are going to produce traits. And actually, these guys are producing a lot of traits. We currently have a sorghum TD of more than 900 variables. But I don't think that it is a good step here to bring it to here, because there is a lot of crap here. The people are not really concerned about sharing, they just describe like this. So I think the way to do is to publish the data, and once it's ready, peer-reviewed, we send it to the crop ontology. The second way to do that is I think we can have direct contribution from the communities. So there are different communities working on sorghum. One of them is the Yavao group, which had the expectation to set up multi-environment trials in West Africa. So what we did is that we picked 9 of them, we unlocked them for one day in a room, and some of them already contributed, so they were guilty to have contributed to the previous version. And we make them think about what they needed to set up this experiment. So they validated that 14 were okay, but they needed to add 13 additional variables. So this, we can do that all over the world, as there are other communities working on sorghum. But the main thing here is that it's only when people get involved that they are going to need to measure traits and share with colleagues that the process of describing traits is really efficient. The second thing we need, but we have it, I think, we have Git-like tools to interact. And what I think, I already work with the guys involved in sorghum, but I think we really need people that are concerned by the species who will curate the data, and who know what there is in the trait dictionary, and who put the fits in the field often to be able to challenge the people who want to add new traits. And the last thing, I think, to be able to take advantage of ontologies is really to have the editor support. So I'm not going to learn you that science are papers, that we can increase the quality of science through ontologies. But I think as a community here, as reviewers, we need to contact the editors, and each time that you receive a paper, you need to ask, is this trait located in an ontology, and how you describe it? Otherwise, nobody is going to use your information because it's not useful, it's not described. So we really have something to do here at the end of this Congress, I think, to contact the editors. Last slide is just to, okay, it's a collaborative effort, of course. So the main guys who contributed to that are Jean-François. He sits at the front at the end of the room. So he's in the story of the crop of the sorghum ontology since the beginning. Sandrine is also leading the PhenoHarmonies organization, and she's contributing to the database development to store our traits. I want to acknowledge the Yavao group who contributed to that, and also, of course, Elisabeth and Marie-Angélique, not only for organizing this event, but also to being able to have the patience to help us to enter the data in the crop ontology. That's it. Thank you.